Estimation of fat, dear students, is another analytical method that is very routinely performed for proximate analysis of food. While we do this analysis, there could be different types of analysis, like it can measure the total amount of lipids in the food, or it can measure the types of lipid present in a specific food, or thirdly, it might look at the physical and chemical properties of these lipids. So these three kinds of analysis are normally performed: amount of lipid present in the food, the type of lipid in the food, and the physical properties of the lipid in the food. And why this analysis is important? I think the four most important thing is the nutritional value of fats, such as cholesterol content in the fat, or maybe the good things like omega-3 present in fat contents or fat soluble vitamins, because they have got very important nutritional significance for human nutrition. So this is an importance why we focus on this analysis. Then oxidative stability is another criteria that would determine like how much shelf life a lipid would have. So in order to determine this oxidative stability, we might perform certain food analysis. And another reason is the physical properties of food, such as melting point, or other physical properties as well. They would determine the quality of the end product, such as chocolates and margarine. We want to have a specific melting point, and that melting point defines the quality of those products. So this is why it is important to do fat analysis. When it comes to sample collection or preservation of food for which we do the fat analysis, it can depend upon the type of food. Like, for example, in certain instances we might have a liquid food such as milk from which we want to extract fat, or it could also be a solid food which might have a little amount of fat that need to be extracted. So that would define what kind of sampling we need to do and what kind of preservation of that sample is needed. Similarly, the other natures like volatility of the fat or the physical attributes of fat, that would also determine like how we are going to draw sample from food. Type of analytical procedures, there might be different analytical procedures. So we need to look at what kind of analytical procedure we are following, and that would dictate you like what kind of sample or how much of the sample you are taking. Or if you are not performing this analysis immediately, then how would you going to preserve that fat sample before it is actually analyzed in the lab? So there could be certain reasons for food analysis, like there could be economic reasons why we do this fat analysis, because certain times this amount of fat present in food would determine the price of the final product. It could be legal. For example, the countries would have their legal standards for certain food products to have a specific amount of fats or specific properties of fat present in that food. So it has to be performed, this analysis has to be performed to determine the legal compliance of food product with the country standards. Health is another very important and significant factor for why we do this analysis, because there are certain things in fats which are related to good health, such as omega-3 or fat soluble vitamins, or unsaturated fatty acids. And similarly, there could be other things which may not be very healthy, like cholesterol content, saturated fat contents. So health is one of the important reasons why certain times we have to do this fat analysis. Quality of the food product or quality of the fat, actually important factor, again why we do this analysis. Processing of fats and vegetable oil or vegetable ghee, etc., has to be dependent upon, again, the quality of the food product or the characteristics or attributes of that fat product. So for example, in order to process certain products or in order to achieve the desirable end product, we need to know certain characteristics of food, before it actually goes into the processing process. When it comes to the methods of analysis, one of the most important element to consider is what is the solvent that we are going to use, because all the methods of analysis of fat contents are dependent upon using a solvent, and that solvent would extract that fat out from the food sample, and then that fat extracted is weighed and expressed as percentage or in terms of mass, like how much fat is present in a specific food. Certain food contents would need to be pre-treated before doing actual analysis, because in certain foods maybe the particle size is too high. We need to reduce that particle size to facilitate the removal of fat with solvent, and similarly we need to also dry certain samples to reduce the amount of moisture present in food that would otherwise interfere with the removal of fat contents, and there might be error in the final results if we don't do these to pre-treatment of samples. If you look at the good attributes of the ideal solvent that we use for fat extraction from the food, it should have high solvent power for extraction of food. Similarly, it should have low solvent power to extract non-lipid components of the food, and it should have no residue. It should not evaporate very differently. It should be easily evaporated from the food, so that we can actually remove that this solvent easily. Similarly, it should not be flammable, and it should have lower boiling point. So these are some of the factors that need to be considered for selection of solvents, because there are a variety of solvents available in the market, and we need to decide which solvent we are going to use for our particular analysis. One of the most widely used method is Soxhlet method. It consists of Soxhlet extracting apparatus in which we actually cover the food with some of the solvents such as ethyl ether or diethyl ether that would actually remove the fat contents from the food, and then this is a continuous process. Once this solvent removes the fat content, it is evaporated off again, condensed, and it goes, after condensation, it goes back to the food. It removes certain more amount of fat from the sample, and this method continues unless we have removed all the fat present in a food. Once we are left with this solvent and fat in the flask, then we evaporate off that solvent, and the remaining amount of fat in the flask is actually measured gravimetrically, and that would tell us about the amount of present that was in the food. So that can be determined, and this is how we actually do fat analysis, total fat analysis for different foods.